In the previous video, we discussed the signal transduction hypothesis of adaptation when we exercise. This was a general overview to give you an understanding of the concept. We didn't go into specific detail, but one of the steps highlighted the signal transduction pathways that help to convey and compute the sense input. When we look at the long-term adaptation to exercise in skeletal muscle, we established that it is a complex process involving specific signaling mechanisms. The impacts of these changes are determined by factors such as the amount, intensity, and regularity of training, along with the lifespan of the protein. Furthermore, the details of the adaptation response are specific to the types of exercise stimulus. Muscle contractions during exercise trigger temporary increases in messenger RNA. These mRNA molecules carry genetic information from DNA to the protein building sites known as ribosomes. For many genes, this increase in mRNA typically reaches its peak 3 to 12 hours after exercise and returns to normal levels within 24 hours. The rise and fall of mRNA levels generally coincide with the production of proteins during the adaptation process, as the body adjusts to a new steady state. Therefore, regular exercise sessions lead to increased gene transcription activity and subsequent protein synthesis. This suggests that the long-term adaptations to training likely result from the cumulative effects of each short exercise session, altering the steady state levels of specific proteins and establishing a new functional threshold. Long-lasting endurance training induces a range of metabolic and morphological changes in skeletal muscle. These changes include the generation of new mitochondria, the cellular powerhouse responsible for energy production, transformation of muscle fiber type, and alterations in the body's metabolism of energy sources. On the other hand, heavy resisted exercise stimulates adaptive mechanisms that lead to muscle hypertrophy and increased force production during contraction. These diverse exercise modes yields drastically different functional outcomes, and the genetic and molecular mechanisms of adaptation involved are distinct. Each exercise mode activates or suppresses specific pathways and subsets of genes. Consistent training in each mode leads to unique exercise-induced adaptations in muscle fibers. Therefore, aerobic endurance and heavy resistance training can be viewed as two extremes along an adaptation continuum. In subsequent modules, we will explore the molecular responses to both exercise types. To aid our understanding, this video will highlight and briefly discuss some of the main primary and secondary messengers involved in the signal transduction pathway. It is important to note that this video provides an overview rather than an in-depth explanation of each messenger, but it will assist in further learning as we delve into different modes of exercise. Converting a mechanical signal generated during muscle contraction into a molecular event that promotes muscle cell adaptation involves the increased activity of primary and secondary messengers. These messages initiate a cascade of events resulting in the activation or suppression of specific signaling pathways, which regulate exercise-induced gene expression and the balance between protein synthesis and degradation. Looking at primary messengers, understanding the exact mechanisms that enable skeletal muscle cells to interpret and respond to contraction has been challenging due to the complexity of the process. It involves proposed mechanoreceptors which link neuronal, mechanical, and biochemical events, and the many cellular candidates acting as potential primary messengers that convert the mechanical signal. It's also believed that these primary signaling messengers do not act alone, but rather result in a complex, multi-layered signal involving redundancy and crosstalk. Despite these complexities, several potential messengers have been identified, including mechanical stretch, calcium flux, redox state, and phosphorylation state. When skeletal muscles are contracted during exercise, the muscle fibers are physically stretched. This mechanical stretch is thought to act as a signal that triggers several pathways within the muscle cells, leading to the activation of specific signaling cascades. For example, mechanical stretch has been shown to activate pathways such as calcineurin, mitogen-activated protein kinase, that's MAPK, and insulin-like growth factor, IGF. These play a role in muscle growth and adaptation. These pathways are believed to activate the transcription of genes related to muscle function and adaptation, ultimately leading to the synthesis of proteins that alter the structure and function of the muscle in response to exercise. Calcium plays an important role in the contraction of skeletal muscles. When a muscle fiber is stimulated, calcium ions are released from the sarcoplasmic reticulum, which is a specialized storage compartment within a muscle cell, and it's released into the muscle cell cytoplasm. This surge in calcium triggers muscle contraction. Calcium ions are believed to act as a messenger in the cell, with changes in their concentration initiating signaling pathways that lead to muscle adaptation. For example, they activate certain proteins that promote transcription, the first step in protein synthesis. During exercise, the muscle's demand for energy increases. This intensification of metabolic processes leads to the production of reactive oxygen species, ROS. These molecules are unstable and highly reactive due to the presence of unpaired electrons. Although reactive oxygen species can be harmful to cells at high concentrations, they are also necessary for normal cellular function, including acting as signaling molecules that modulate several signaling pathways. The redox state of the cell, which represents the balance between reactive oxygen species and antioxidants, is thus a potential regulator of exercise-induced adaptations in skeletal muscle. Here is when muscle contraction during exercise demands energy, which is provided by the molecule adenosine triphosphate, ATP. The use of ATP during exercise leads to an increase in adenosine monophosphate, AMP levels within the cell, thereby increasing the AMP to ATP ratio. This change in the cell's energy state is a signal that activates several signaling pathways, including the 5-adenosine monophosphate activated protein kinase, or AMPK pathway. AMPK is an energy sensor in cells, and when activated, it initiates several processes such as glucose uptake and fatty acid oxidation to produce more ATP. It also promotes the synthesis of new proteins, leading to muscle adaptation. After the initiation of the primary signal, additional kinases or phosphatases are activated to mediate the exercise-induced signal. Mammalian cells contain numerous signaling cascades that are highly regulated and interconnected, forming a complex and sensitive transduction network. This network allows for efficient translation and integration of incoming signals. In this section, we will focus on the adenosine monophosphate activated protein kinase, that's the AMPK mediated signaling, and also the insulin-like growth factor signaling pathway. It's also important to note that this list is not exhaustive. However, it will give you a good understanding of the primary function of AMPK in this case. The role of AMPK in skeletal muscle has garnered significant scientific interest. In simple terms, AMPK is directly activated by AMP, that's adenosine monophosphate, which is a molecule related to energy regulation and is sensitive to changes in the cellular AMP to ATP ratios. When cellular energy levels are depleted, such as during skeletal muscle contraction, AMPK is acutely activated to both conserve and generate ATP. Once activated, AMPK performs several functions. It can increase ATP production by promoting glucose uptake without the need for insulin, and it enhances the breakdown of fats. AMPK also influences muscle adaptations by modifying protein production and gene expression. It is associated with the regulation of gene expression related to fatty acid breakdown in mitochondria and dampening protein synthesis by reducing the activity of the insulin growth factor pathway. Research suggests that the AMPK activation varies depending on factors such as exercise duration, intensity, and specific muscle fiber types. It has been observed that exercise leads to an increase in AMPK activity, particularly in the red oxidative muscle fibers, which are more resistant to fatigue compared to white glycolytic, more fast-twitch fibers, which are more prone to fatigue. Since AMPK is considered as this energy sensor, its activity has been found to increase during submaximal aerobic exercise, higher treadmill speeds, and greater cycling power. Interestingly, the state of muscle adaptation appears to influence the AMPK response. Regular aerobic training reduces the acute AMPK response to long-term and intermittent submaximal exercise, but the response is preserved if the exercise is sufficiently challenging. In the context of resistance training, the role of AMPK is less understood due to the limited research. However, some studies suggest an increase in AMPK activity following resistance exercise. This increase might be related to enhanced glucose uptake independent of insulin. Nevertheless, the physiological significance of AMPK increases after resistance training requires further confirmation. Comparisons between endurance and resistance training regarding AMPK responses are scarce. Animal models provide some evidence suggesting that AMPK activation differs depending on whether the exercise is more endurance-like or resistance-like. For example, athletes who switch their regular training routines may experience increased AMPK activity, suggesting that the nature and novelty of the exercise stimulus can alter the AMPK response. Given that long-term endurance and resistance training lead to different functional outcomes, AMPK signaling may play a key role in these distinct adaptations. Now looking at the insulin-like growth factor signaling pathway, this pathway plays a pivotal role in regulating muscle health and growth, encompassing essential processes like muscle breakdown, proteolysis, muscle growth, hypertrophy, glucose uptake, glycogen storage, and cell growth and differentiation. Insulin-like growth factor 1, IGF-1, is an important part of this pathway, and its importance in muscle has been demonstrated in several different experimental models. During muscle contraction, the muscle releases IGF-1, which then acts as a local growth factor by binding to its receptor on the muscle cell membrane and starting a chain of molecular events. We will focus on two important components of this pathway. The first is AKT and its associated substrate targets. AKT is a protein that plays multiple crucial roles in our cells. It comes in three different forms, referred to as isoforms. Two of these, known as AKT1 and AKT2, are mainly found in skeletal muscle. These two isoforms are thought to have different functions. AKT1 is believed to be primarily associated with muscle hypertrophy, whereas on the other hand, AKT2 appears to be involved in the process that enables glucose, our body's main energy source, to enter our cells. This process is known as glucose transport. Before AKT can perform its functions, it must be activated. This activation process involves a series of steps, including the movement of AKT to the cell membrane and its modification by other molecules in a process known as phosphorylation. This phosphorylation happens at specific sites on the AKT molecule and is necessary for its full activation. Once activated, AKT can interact with various target molecules, influencing a range of biological processes. Here are some of the key ones. The first is protein synthesis. This is the process by which new proteins are made in the cell. AKT enhances protein synthesis through interactions with molecules such as mTOR, mammalian target of rapamycin, and the tuberous sclerosis complex, TSC, and the GSK, or the glycogen synthase kinase 3. Looking now at mTOR, this is a protein complex that plays a significant role in the body and coordinates various cellular processes. It acts as a master regulator, integrating signals from growth factors, nutrients, and energy status to control cell growth, metabolism, and protein synthesis. As a protein that, when activated, can stimulate the production of proteins, thereby contributing to muscle growth. mTOR exists in two main configurations, or protein complexes. One of these, known as the mTOR-RAPTOR complex, involves mTOR partnering with a protein known as G-beta-L and another one called RAPTOR. This specific formation enhances cell growth. The other configuration involves mTOR binding with the G-beta-L protein and another protein called RICTOR. This formation, the mTOR-RICTOR complex, is important for the activation of another protein, AKT, and for the managing of the actin cytoskeleton. This is the framework of filaments within the cell that gives us the shape and enables it to move. The activity of the mTOR-RAPTOR complex is regulated by proteins upstream in the signaling pathway. One of these proteins is Reb, a G protein that directly interacts with mTOR and activates its function. The TSC complex, which consists of TSC1 and 2, plays a role in regulating the mTOR pathway. It does this by affecting the activity of the Reb protein. In a way, the TSC complex acts as a brake on mTOR, signaling by inhibiting a protein called Reb. When TSC1 and 2 are active, they work together to enhance the inactivation of Reb, preventing its ability to activate mTOR. This keeps protein synthesis and cell growth in check. However, when growth factors like IGF-1 and nutrients are present, signaling pathways activate AKT, which phosphorylates and inhibits the TSC1-TSC2 complex, thereby relieving the inhibition on Reb. This AKT-mediated phosphorylation of TSC2 releases the brake on Reb. As a result, Reb becomes active and can stimulate mTOR signaling and initiate protein synthesis and cell growth. The downstream targets of the mTOR-RAPTOR complex includes P70S6K, 4EBP1, and EIF4B. These are all involved in mRNA translation and protein synthesis. Activation of these targets by mTOR leads to an increase in the production of proteins essential for cell growth and other cellular functions. In experimental studies, mTOR has been found to be responsive to mechanical stimuli in muscles. Overloading the muscles or subjecting them to high-frequency electrical stimulation activates mTOR, resulting in increased protein content and muscle hypertrophy. However, the response of mTOR to muscle contractions appears to be intensity-dependent, as low-frequency stimulation does not produce the same effect. This suggests that mTOR's responses are linked to the intensity of muscle activity. Interestingly, voluntary wheel running and resistance training in animal models have been shown to increase mTOR activation. Although traditionally associated with muscle growth, recent evidence suggests that mTOR might also play a role in managing the functionality of mitochondria, the cellular powerhouse responsible for energy production. Therefore, this dual role of mTOR in both protein synthesis and mitochondrial function adds complexity to its involvement in exercise adaptation. The increase in mTOR activation following resistance training aligns well with our understanding of mTOR's role in enhancing protein synthesis when muscle hypertrophy is induced. In human studies, both a single bout of resistance exercise and regular resistance training has been found to increase mTOR activation. These findings support the notion that mTOR is an important player in the anabolic processes stimulated by resistance training, contributing to muscle growth and adaptation to exercise. In summary, mTOR is a versatile protein complex that integrates signals from growth factors and nutrients to regulate cellular processes. Its different complexes such as mTOR-Raptor and mTOR-Rictor play distinct roles in cell growth, protein synthesis, and metabolism. The regulation of mTOR activity by upstream proteins like Reb and the TSC1 and TSC2 complex further fine-tunes its functions. Understanding the complex interplay of mTOR in response to various stimuli and its role in exercise adaptations is an ongoing area of scientific research. The AKT-FOXO or FOXO pathway is a signaling pathway involved in the regulation of gene expression and cellular growth and survival. It primarily revolves around the interplay between AKT, this is protein kinase B, and FOXO, fork head box O, transcription factors. One of the key actions of activated AKT is phosphorylating FOXO proteins, which leads to their inactivation. FOXO proteins are transcription factors that regulate the expression of genes involved in various cellular processes, including cell cycle control, apoptosis, and stress response. Upon phosphorylation by AKT, FOXO proteins undergo a change in their cellular localization, or in other words, the addition of phosphate groups to FOX proteins by AKT leads to their movement or relocation within the cell. They are exported from the nucleus to the cytoplasm, making them unable to activate the expression of target genes. FOXO proteins, when active and in the nucleus, bind to specific DNA sequences and promote the expression of target genes involved in processes such as cell cycle arrest and apoptosis. By inhibiting FOXO through phosphorylation, AKT prevents the activation of these genes, allowing cells to continue growing and avoiding cell death. This promotes cell proliferation and survival, contributing to the growth and maintenance of tissues. GSK3 is another protein that can influence the process of building new proteins in our cells. GSK3 plays a role in controlling the start of protein synthesis by affecting another protein called EIF2B. Think of EIF2B as a key that unlocks the door to protein production. When GSK3 is active, it adds a chemical tag called a phosphate group to EIF2B, which acts like a lock, preventing it from efficiently carrying out its job. This slows down the initiation of protein synthesis. However, when GSK3 is deactivated, it doesn't add the phosphate tag to the EIF2B anymore. This allows EIF2B to function freely like an unlocked door and initiate the process of protein synthesis, leading to the production of more proteins. In simple terms, GSK3 can control protein synthesis by affecting EIF2B. When GSK3 is active, it puts a lock, slowing down protein production. When GSK3 is inactive, it removes the lock, enabling EIF2B to initiate protein synthesis, resulting in increased protein production. AKT also has a role in glycogen synthesis and therefore plays a role in the transport of glucose into cells, thereby affecting energy synthesis. Referring to GSK3 we just discussed, the role that AKT activation plays in the inhibition of GSK3, which results in increased protein synthesis. With respect to glycogen synthesis, this same process of AKT activation, which inhibits GSK3, allows another enzyme called glycogen synthase to do its jobs, that is glycogen synthesis and produce glycogen. Whilst we are discussing glycogen synthesis, AS160 is another protein that AKT interacts with. AS160 is a protein that plays an important role in the regulation of glucose uptake into cells. When AKT is active, it phosphorylates. That adds a phosphate group to AS160. This phosphorylation event releases AS160's hold on another protein called GLUT4 or GLUT4. And this is a glucose transport protein and responsible for bringing glucose into cells. Therefore, translocation of GLUT4, regulated by AS160, allows it to move from intracellular compartments to the cell membrane in response to signaling cues. When GLUT4 reaches the cell membrane, it facilitates the uptake of glucose from the blood into the cell. The relationship between exercise and the activity of AKT, that key protein in skeletal muscle, is a subject of ongoing research, and there are different opinions and findings about it. Specifically, it's unclear whether AKT activity increases or remains constant in response to exercise. Some researchers have also explored how different types of exercise affect AKT activity. A few studies have demonstrated that cycling for 30 to 60 minutes at either low intensity, that's at around 70% of maximal oxygen uptake, or high intensity at around 125% of VO2 max, was enough stimulus to increase AKT phosphorylation. Conversely, another study reported that there was no change in AKT activity 15 minutes after completing an hour of cycling at 70% of VO2 max. Other studies have shown an increase in AKT activity after resistance training, while some have found no response from AKT in this exercise mode in human subjects. Other work has also reported that AKT activation varied with different types of exercise, that's endurance and resistance, in athletes with different training histories. In summary, authors have commented that we still have a lot to learn about how exercise influences AKT activity. Factors such as the type and intensity of exercise, the timing of when muscle tissue samples are collected, and the specific responses of different AKT isoforms could all contribute to these varied findings. Differences in experimental design, including the use of mixed fiber type samples, might also explain some of the discrepancies in the research findings. Despite these inconsistencies, there is strong evidence that AKT plays an important role in how skeletal muscle adapts to exercise. However, identifying and verifying the specific roles of the different AKT isoforms using live exercise models continues to be a challenge. Resistance training, which typically increases muscle tension and growth, and endurance training, which increases the use of energy sources in the body, both seem to increase AKT activity. Considering that AKT is believed to regulate protein synthesis and glucose transport, this increase makes sense. Therefore, AKT seems to be a major player in guiding some specific adaptations to both endurance and resistance training in skeletal muscle. That's all for this video. In the next video, we will look at some of the molecular responses to endurance exercise. Again, this won't be in massive detail due to the depth of this area, but rather it will give you an overview of the different molecular responses associated with different exercise types.